Hello everyone. Let's start with the beta oxidation of saturated fatty acids. So here we are going in details with the oxidation of fatty acid, and we are going to take palmitic acid as one of the example regarding the generation of number of ATP molecules during the oxidation of the fatty acid and the amount of the energy that is being generated by this palmitic acid. So let's start. Now regarding the basic introduction, so we all know that the fat, they are the important source of energy in our body. Around 1 gram of fat, it gives approximately 9 kilocalorie energy in our body. Moreover, they remain as a triacylglycerol or triglyceride in the stored form. Generally, it is found in the adipose tissue in around our, mostly it is found in around our abdomen. Moreover, they constitute about 84% of stored energy. Now, if we again go into the basic of beta oxidation. So, beta oxidation, what is beta oxidation? Simply, it is the oxidation which can be defined as the oxidation of fatty acid at beta carbon atom. First of all, at beta carbon atom. Next one, this beta oxidation is generally results in the sequential removal of two carbon fragment acetyl CoA. This process, it is a completely aerobic process and it occurs in the mitochondria, and moreover after the production of the acetyl CoA, it is directly fed into the Krebs cycle for further processing. Now again, if we see in the basic introduction, so first of all, where the oxidation of this fatty acid takes place, the oxidation of this fatty acid generally takes place in three important organs, that are the liver, kidney and the heart. But preferentially fatty acid oxidation, it don't take place in the brain. Now there are different types of oxidation. So basically we have the beta oxidation, alpha oxidation, along with that we have omega oxidation. Now again, if we see what actually happens in the beta oxidation, so basically beta oxidation involves three steps. First one, it is the activation of the fatty acid in the cytosol, of the cell, in the cytosol. The next step, it is the transport of this activated fatty acid which has formed in the cytosol into the mitochondria. This is known as the carnitine shuttle. And finally, the beta oxidation proper, means the basic steps of the beta oxidation, takes place in the mitochondrial matrix. So now the activation of fatty acid, Fa. So how it is going to get activated in the cytosol? So to get activated, this will be proceeded with a particular enzyme. This is known as fatty acid kinase, also known as the Acyl CoA synthesis. It is present in the cytosol. Apart from its activity in the fatty acid oxidation, it takes place in other metabolic reactions also. Now this thiokinase, actually it needs the ATP molecule as well as coenzyme ASH as a cofactor or coenzyme. Along with that, it needs the magnesium. Now once they are going to react, the product of this reaction is the fatty acid acyl CoA will get formed along with water. To summarize this reaction, we can check here that the fatty acid, it combines with the coenzyme A. Along with that, it will utilize ATP molecule. Now the final product will be the fatty acid CoA. Along with that, AMP, adenosine monophosphate, will form along with two molecules of inorganic phosphate. Okay, now the next step, it is the transport of fatty acyl CoA which has formed the cytosol. Now how we are going to transfer it within the mitochondria. That's an important step here. So here you have to remind something in your mind. You have to keep something in your mind. First of all, the inner mitochondrial membrane is impermeable to fatty acid. I repeat, the inner mitochondrial membrane is totally impermeable to fatty acid. Thus, to transport this fatty acid into the mitochondria, we need to have one specialized carrier system. So this carrier system, it is also known as the carnitine carrier system or carnitine shuttle, so which will operate to transport this activated fatty acid, whatever it has formed, that is the fatty acid CoA in the cytosol. It will be now transported from the cytosol into the mitochondria. But you have to keep one thing in your mind, that the long chain acyl CoA, they generally traverse the inner mitochondria membrane with a special transport mechanism. This is known as the carnitine shuttle, which I already described. So this is the molecule of the carnitine. You see here there are two enzymes. How they are going to perform in the transport, I am going to discuss in my second slide. Okay, now let's discuss on the transport of acyl CoA into the mitochondria, which is considered as the rate limiting step. Rate limiting step because it is the most crucial step in the oxidation of fatty acid. It is going to control all the other steps, or basically the rate of the fatty acid oxidation. So it generally takes place in the four basic steps where enzymes, different types of enzyme it works. You can see in the figure. So first of all, the first crucial step, it is the acyl group. From the acyl CoA, whatever it has formed, from the CoA it is transferred. Now it will get transferred to the carnitine to form acyl carnitine. It will now form acyl carnitine. Acyl CoA, it will now form acyl carnitine, and it is catalyzed by a specific enzyme known as carnitine acyl transferase 1. And you remember one thing, this step, it takes place outside the mitochondrial membrane. It is generally takes place outside the mitochondrial membrane. Now the next step is that this acyl carnitine, whatever it has formed now, it will be now shuttled, or you can say it will be now transported across the inner mitochondrial membrane by a enzyme. This enzyme, it is known as the translocase enzyme. Trans means transport, translocase enzyme. The third step, it is the acyl group. From this acyl carnitine, this acyl group, now it is transferred back to the coenzyme CoA in the matrix by the carnitine acyl transferase 2. It is the second enzyme. I repeat this once again. The acyl group, from this acyl carnitine, now it is transferred back to the coenzyme A, which is present in the matrix of the mitochondria, and it is done by a specific enzyme known as carnitine acyl transferase 2. And finally, this carnitine, the remaining part, as because acyl it has been utilized, the carnitine it is remaining, this carnitine it will now return back to the cytosolic side, to the outer side, and this will be done, or this will be helped by again the enzyme that is known as the translocase enzyme. It will be exchanged, or it will be sent in exchange for an incoming acyl carnitine. Means this carnitine it will go out, and one more molecule of acyl carnitine it will come in. Okay, now what will happen once this fatty acid it will get inside the mitochondrial matrix? We know that the beta oxidation it will take place in the mitochondrial matrix, but what are the steps? So basically the beta oxidation it takes place in four distinct steps. These are the steps. The step 1, it is the oxidation by FAD link dehydrogenase. Oxidation will be done. The step 2, it is the hydration. It will be done by the enzyme hydratase. The third step, it is the again the oxidation. It will be done by NAD link dehydrogenase enzyme. The fourth step, it is the Thiolytic cleavage. It will be done by the enzyme Thiolase. So basically, the four step include oxidation, hydration, again oxidation, and finally the Thiolytic cleavage. The enzymes responsible are the FAD-linked dehydrogenase, hydratase, NAD-linked dehydrogenase, and thiolase. Okay, step 1, that is the oxidation by FAD-linked dehydrogenase enzyme. So here we can see I have given you the structure of palmitoyl coenzyme A. Now how it will undergo oxidation. So basically what happened here, the first reaction, it is, I already told you, it is the oxidation of acetyl coenzyme A by an enzyme known as acetyl coenzyme A dehydrogenase, which will now, after the oxidation, will give you alpha beta unsaturated acetyl coenzyme A, that which is also known as enoyl CoA. And in this step, the FAD, it becomes the hydrogen acceptor just to oxidize the acyl coenzyme A. So that is, we can also say that the acetyl coenzyme A undergo dehydrogenase by an FAD dependent flavoenzyme, that is your acetyl coenzyme dehydrogenase. A double bond is formed between the alpha and the beta carbon, that is in between the second and third carbon. Again here, in the step 2 involves the hydration by an enzyme known as hydratase. So what actually will happen here, you can see in the slides that it is the hydration of the double bond. I repeat, it is the hydration of the double bond to beta hydroxy acyl coenzyme A. The product, it is also known as 5-hydroxy acyl coenzyme A. Moreover, enoyl coenzyme A, which has formed earlier, that is the enoyl coenzyme A hydratase enzyme. Enoyl coenzyme A hydratase enzyme brings about the hydration of the double bond here. It will bring the hydration of the double bond here to form the beta hydroxy acyl coenzyme A. Now we are going to the third step, the step 3, that is the oxidation by NAD linked dehydrogenase enzyme. So you can see here, so what will happen here, the third reaction, it is the oxidation of beta hydroxy acyl coenzyme A to produce beta ketoacyl coenzyme a NAD dependent reaction. So beta hydroxyl coenzyme A dehydrogenase, the enzyme, it will catalyzes the second oxidation and generates the NADH. You can see in the slides. The product formed is actually the beta ketoacyl coenzyme A. And lastly, step 4. The step 4 is considered as the Thiolytic cleavage, and it is done by the enzyme Thiolase. So briefly, you can see in the slide, the fourth step, it is the cleavage of the 2 carbon fragments. I repeat, it is the cleavage of the 2 carbon fragments by splitting, by breaking the bonds between the alpha and the beta carbon. And this splitting, or you can say cleavage, it is done by the enzyme Thiolase. Here actually, it is the final step of the beta oxidation. So we consider here in this final step of the beta oxidation, it is the here actually liberation of 2 carbon fragments, acetyl coenzyme A from acyl coenzyme A takes place. This occurs, this whole reaction, it occurs by a Thiolytic cleavage, and it is actually catalyzed by beta ketoacyl coenzyme A thiolase, or the thiolase enzyme. That is, don't get confused here. Actually, the thiolase enzyme, it is also known as the beta ketoacyl coenzyme A thiolase. So the reaction I have shown here. Okay, now coming to the analysis of energetics. the last step, that is what will happen when the palmitic acid will undergo beta oxidation, that is amount of the ATP produced. So we know that the palmitic acid is a 16 carbon compound. Now this palmitic acid, it will get converted into palmitoyl coenzyme A to get oxidized. So for the complete oxidation of palmitoyl coenzyme A, we need 7 molecules of coenzyme A as a coenzyme, regarding which I have told you earlier. Moreover, we need 7 molecules of FAD, 7 molecules of NAD. So these are actually, FAD and NAD, they will help in the process of the oxidation. Now they will get reduced, they will help in the process of oxidation. Now the product is, after the reaction, we will get 8 molecules of acyl coenzyme A plus 7 molecules of FADH2 plus 7 molecules of NADH, and moreover we will get 7 hydrogen, that is 7 proton. Now you see here the FADH2 molecules, how many ATP it produce? Generally noted, it produces 2 ATP molecules. Moreover NADH2 or NADH, it produces 3 ATP molecules. So sum total it is 5. So 7 molecules they are involved here, right? You can see in the reaction that 7 molecules of FADH2 and 7 molecules of NADH, right? So therefore this combination, that is 2 plus 3 FADH and NADH, 2 plus 3, that is 5 into 7 times they are occurring, right? It is equal to 35 ATP they are produced, they have produced. Now moreover, the acyl coenzyme A, when it will now enter the citric acid cycle, that is it will undergo oxidation, acetyl coenzyme A, each molecule it will give 12 ATP. So 8 molecules of acyl coenzyme A has produced, right? So 8 molecules into 12 number of ATPs, it is 96 ATP. Thus, but we know that we have utilized 2 ATP molecules during the time of activation of fatty acid, right? If you remember in my previous slides. So the net energy gain, what is the net energy gain? Simple calculation, the amount of the energy which has been produced, moreover the energy which has been utilized. So you can see here 35 ATP it has been produced more from the FADH2 and NADH, right? Moreover 96 ATP has been produced from the oxidation of acetyl coenzyme A in the citric acid cycle. So 35 plus 96 minus 2, that is that these 2 ATP molecules has been utilized during the activation of fatty acid. Thus we get 129 total number of ATP molecules produced from a single oxidation of palmitic acid, which will be done by the process of beta oxidation. So lastly, thank you everyone for your patience hearing. I hope I'm able to make you understand regarding the beta oxidation of fatty acid. Now if you like the video, please press the like icon. You can share the video with your colleagues, with your classmates for their better understanding, and for further notification from my channel you can press the bell icon. Thank you everyone once again.